Homeostatic mechanisms help to regulate blood pressure by maintaining a constant environment. In fact, the word homeostasis itself addresses the body's ability to remain healthy regardless of what is happening and changing in the world around it. There are three main homeostatic mechanisms involved in blood pressure regulation: neural, endocrine, and autoregulatory. Neural systems involve cardiovascular centers located in the medulla oblongata, which regulate vascular homeostasis by managing cardiac output or by adjusting blood vessel diameter. Baroreceptors, mediated by the autonomic nervous system, are specialized stretch receptors located within blood vessels and heart chambers. They integrate the regulation of blood pressure by sending impulses to the cardiovascular center. When blood pressure rises too high, the rate at which the baroreceptors fire increases, triggering parasympathetic stimulation of the heart. This causes a fall in cardiac output and hence blood pressure. Vasodilation of the peripheral arteries also causes a decrease in blood pressure. When blood pressure drops too low, the baroreceptors fire at a slower rate, triggering sympathetic stimulation of the heart. This increases blood pressure by causing an increase in cardiac output and vasoconstriction of the peripheral vessel. Chemoreceptors in the medulla oblongata, carotid and aortic bodies regulate blood pressure homeostasis by monitoring oxygen, carbon dioxide, and pH levels. Impulses from these chemoreceptors are conducted to cardioaccelerator, cardioinhibitory, and vasomotor centers in the medulla oblongata. Decreased blood oxygen, increased carbon dioxide, or decreased pH decreases the parasympathetic stimulation of the heart, increasing heart rate and hence blood pressure. Sympathetic stimulation is increased in response to decreased blood oxygen, increased carbon dioxide, and decreased pH levels, resulting in an increase in heart rate and stroke volume. The endocrine system consists of hormones and catecholamines that interact with the kidneys and cardiovascular system in order to regulate blood volume, and target either a specific population of cells or the majority of cells in order to regulate blood pressure. Epinephrine and norepinephrine are both catecholamines released by the medulla oblongata during the fight or flight response. They cause vasoconstriction of blood vessels to organs not essential in the response and increase heart rate and blood pressure. Antidiuretic hormone is secreted by the hypothalamus in response to increased osmolarity of tissue fluid. It targets cells in the kidney to increase water absorption, help to restore blood volume and pressure. It also causes vasoconstriction of peripheral vessels. Renin is secreted by specialized cells into the bloodstream in response to a decrease in blood flow. Renin, catalyzed by ACE, converts the plasma protein angiotensinogen, angiotensin 1, and then angiotensin 2 in the lungs. Angiotensin 2 stimulates the release of ADH and aldosterone, which in turn increases blood pressure by increasing blood volume. It also increases blood pressure by stimulating the thirst center found in the hypothalamus, prompting an individual to consume more fluid. Furthermore, angiotensin 2 is a very powerful vasoconstrictor of blood vessels, and hence, when catalyzed, significantly increases blood pressure. Autoregulation mechanisms are local and self-regulatory, allowing tissue to adjust its blood flow without stimulation from specialized nervous or endocrine systems. Chemical signals is an autoregulation mechanism that regulates vasodilation or vasoconstriction in precapillary sphincters. The myogenic response, another autoregulation mechanism, is how arteries and arterioles react to an increase or decrease of blood pressure in order to maintain homeostasis.